Hello everyone and welcome to the biology lecture series, and today we'll be discussing some of the practical practice problems from the chapter transportation. So in the previous lecture we have already discussed about the transportation in the higher plants as well as the transportation in the humans. So we have talked about the transpirational pull theory, also known as the transpirational cohesion theory, when we were talking about the transportation in the plants. And then we have talked about the circulatory system, the blood, okay, and the types of blood groups present in the humans when we were talking about the transportation in the humans. Apart from that, we have also studied about the structure of the heart, and we have also studied about the lymphatic system. So in this lecture, we'll be discussing some of the problems that are related to these topics. Okay, so we'll be discussing about some of the objective type questions, or you can say multiple choice questions. All right, so let's start with the first question. The first question is the phenomena of uptake of the water at the expense of energy. So what does this mean? This means the energy is utilized. So there is a process, okay, uptake of the water, and this uptake of the water requires energy, and its energy is obtained by what? By the ATP. So by the breaking down of the ATP, the energy is liberated. ATP, which is known as the energy currency of the cell, this is broken down into ADP and the inorganic phosphate, and in this process energy is released. Now coming back to the question again, now the uptake of water at the expense of energy by the cells and usually against the osmotic gradient. What is osmotic gradient? Means against the concentration gradient. Okay, so whenever you work against the concentration gradient, okay, whenever you work against the concentration gradient, then energy is required, and that is obtained from the breakdown of ATP. All right, let's have a look at the options here. The first option is active absorption, second is the passive absorption, third is the osmosis, and fourth option is diffusion. So let's have a look at each of these options. So active absorption means it requires energy. All right, this requires energy, and energy is liberated by the breakdown of ATP. Okay, so these processes which require energy, these are known as active process. All right, so here the uptake of the water takes place. That's why we are calling this process as active absorption. And active absorption, it happens, it takes place, or what we can say, in the active absorption, there is the movement of the water or the solvent molecules against the concentration gradient. Okay. Now, what is passive absorption? Passive absorption, it doesn't require energy. It doesn't require the energy. Means there is no use of ATP. There is no breakdown of ATP. It is an passive process. Okay, it takes place without the requirement of the energy. Now, what is osmosis? So osmosis is a process in which there is a movement of the solvent particles or the solvent molecules. Generally, water is the most common solvent, and here there is a movement of the molecules from a region of, you can say, higher water potential, or you can say from a region of where of the high or the higher water molecules to the region where there are less or the lower number of water molecules. And here one more thing is required, semi-permeable membrane. So osmosis, okay, takes place across the semi-permeable membrane. Now what is diffusion? Diffusion is the movement of the molecules. Okay, it is the movement of the molecules from where? From a region of higher concentration of the molecules. Okay, higher concentration of the molecules towards the region of the lower concentration of the molecules. And here there is no requirement of a semi-permeable membrane. Okay, so movement of the water is by the process known as osmosis. And if we talk about the exchange of the gases, this exchange of gases takes place by the process known as diffusion. All right, so you can see the difference here. Here we are talking about the solvent molecules in osmosis, and here we are talking about the solute molecules. Okay, now what should be the answer here? Here the uptake of water, it's saying at the expense of the energy and against the osmotic gradient. So here energy is required. So what should be the answer here? Active absorption is the answer here. It is an active process, means it requires energy and it is happening against the concentration gradient. Now next question is, water will be absorbed by the root hairs when? Options are, concentration of solutes in the cell sap is high. Next option is, plant is rapidly respiring. What is respiration? Respiration is the oxidation of the respiratory substrate such as the glucose. All right. So what is happening? Glucose, which is C6H12O6, this is oxidized, and this what happens in respiration? There is a release of the carbon dioxide plus energy is generated by the oxidation of the glucose molecules. Low glucose molecule is a respiratory substrate here. Next option is, they are separated from the soil by a permeable membrane. And the option D is, concentration of salts in the soil is very high. Now for the uptake of the water by the root hair, what should happen? The concentration of the solutes in the cell sap is high. This is the first option. All right, so let's take understand this by this figure. So this is the cell of the root hair. Okay, if the solute potential is very high here, or we can say this, or the cytoplasm or the cell sap here, it is highly concentrated. It is highly concentrated means it has very high number of solute. It has high concentration of solute particles. Okay, then what will happen if the solute potential is high, or you can say if there is high concentration of the solute, then what will happen? Here the water potential will be less. Okay, so cell sap will has less, or you can say lower water potential than its surrounding. Water potential than the surrounding. Okay, now you know water flows from high water potential to low water potential. High water potential means here the concentration of the water molecules is high. The water always flows from high water potential to lower water potential. So here you can see inside the cell, means in the cell sap, due to the high concentration of the solute, the lower water potential develops. here inside the cell you have lower water potential because of the presence of high molecules of the solute. So if the solute potential is high, then the water potential will be less. So what will happen here? In the surrounding, okay, in the surrounding of the cell, you have high number of molecules of water, or you have higher water potential. So water will travel inside. Okay, so this is how uptake of water takes place. All right, and this process, what will you call it? This is known as endoosmosis. Here the water will move inside the cell. Okay, high water potential means higher concentration or higher number of water molecules. So when there will be higher number of water molecules, when the solute will be less. Okay, so they are inversely proportional, you can say. Here means lower number of the water molecules. Okay, this is clear. So what should be the answer here? Answer is concentration of solutes in the cell sap. One of the option was the presence of the permeable membrane. So that was also not correct, because for the uptake of water, which happens by the process of osmosis, it requires a semi-permeable membrane. And the option that were respiration, that is also not correct. Okay, respiration is not involved in the uptake of the water. Clear? Now let's move on to the next question. So here, the root cap has no role in water absorption. Why? So let's have a look at the options here. First option is it has no direct connection with the vascular system. Okay. Next option is it has no cells containing the chloroplast. Okay. Next option is it has no root hairs. And option D is it has loosely arranged cells. So root cap is the protective covering and it protects the root apical meristem. So what is the role of the root cap? The root cap, its role is to protect the growing root. Protect the growing root tip. So you know, whenever the root grows downwards inside the soil, so root caps protect the growing root tip. Otherwise, it will get damaged because of the mechanical or the physical agents or the pressure in the soil. The root cap protects the growing root tip. And there are no root hair present on the root cap. Okay, and the root hair, they are involved in the absorption of the water. They increase the surface area for the absorption. Okay, they are involved in absorption of water. And they also, this is the one point. Another point is they also increase the surface area for absorption. This is clear. Okay, so what should be the answer to this question? Root cap doesn't have any root hairs. That's why it is not involved in the absorption of the water. It only provides protection to the growing root tip. Explanation for this question: the root cap, it serves as a protective layer of the cells, shielding the meristem. Which meristem? Root apical meristem. Root apical meristem from the abrasion and the harm while the root tip advances through the soil. Now, unlike the root hairs, which enhance the water absorption by enlarging the surface area, this I have already explained you, to facilitate the osmosis. The root cap does not participate in the water absorption. So hence the accurate statement will be it lacks the root hairs. Okay, this is clear. Now let's move on to the next question. Now, which of the following is used in measuring the transpiration? So what is the method for measuring the transpiration rate? First option is photometer, second option is cobalt chloride paper, third option is bell jar, and fourth option is none of the above. Now let's have a look at the answer here. So answer is photometer. The photometer is the device which is used to measure the transpiration. Now what is the principle here? The principle relies on the concept that loss of the water equals the water absorbed as indicated by the displacement of an air bubble inserted into the photometer. So there is we use the displacement of the air bubble to measure this rate of transpiration. And what is the concept on which it is based on? It is based on the concept that the loss of water is equal to the water which is absorbed. All right. The photometer is the instrument used for the detection of the respiration rate. The next question is translocation of solutes primarily takes place through. So whenever we talk about the transport of solutes, okay, or movement of solute, then we use the term translocation. And this happens with the help of phloem. Okay, so phloem is involved in the translocation of solutes. So what are solutes? Solutes are the organic substances or the organic molecule, or you can simply say it as the food that is produced by the plants. So let's have a look at the options here. Option is phloem, second option is xylem, third option is cortex, fourth option is pith. So I've already given you the answer. Phloem is the correct answer here. The phloem, you know, it is made up of sieve tubes, companion cells, phloem fibers, and phloem parenchyma. Okay, so basically, sieve tubes as well as the companion cell, these are involved in the translocation of the solutes. Okay, xylem is involved in the transportation of the water. Okay, and xylem is again made up of tracheids vessel, xylem fibers, and xylem parenchyma. So here tracheids and vessels are the main cells through which majority of the water transport takes place. Now what is cortex? Cortex is the outer layer, okay, of, you can say, stem or a root. All right, that is just present beneath the epidermis. It is present beneath the epidermis. Okay, this is just present beneath the epidermis. And pith is the central region present in the stem. All right, so what should be the answer here? Phloem is the answer. And what is the explanation? Now the nutrients synthesized by the leaves are transported as solutes through the phloem tissue to different parts of the plant for storage or for consumption. Now this process is known as translocation. Clear? Now explanation for the rest of the options, why the rest of the options are not correct. So cortex, I've told you, it is a layer located outside the vascular bundles in a stem or a root of a vascular plant, which is situated beneath the epidermis. Okay, so if this is the epidermis. If we talk about a stem or a root, the outermost layer is the epidermis. Okay, so these are the cells of the epidermis. And then beneath the epidermis, you have the cortical cells. Okay, so cortex is present beneath the, so this is the cortex. So it is present beneath the epidermis, okay, but outside the vascular bundle. So vascular bundles, these are present in the center. Okay, so these are the vascular bundle. or you can say the vasculature, which consists of the xylem and the phloem. So it is present above the vascular bundles and beneath the epidermis. Okay. Now pith. Pith is a tissue in the stem of the vascular plants, and pith is composed of soft, spongy parenchyma cells which store and transport the nutrients throughout the plant. Okay, so they are present in the pith of the stems. Clear? Now let's move on to the next question. The mature human erythrocyte. What are erythrocytes? RBCs, red blood cells. Okay, has the typical characteristic of, the options are eukaryotic cell, prokaryotic cell, both eukaryotic or prokaryotic cell, or neither eukaryotic or prokaryotic cell. So you know RBCs, they are present in humans and humans are eukaryote. But what happens? RBCs, if we talk about the RBCs, they lack the nucleus. They lack nucleus. So whenever they mature, then they lose the nucleus. Initially, the nucleus is present, and when in mature RBCs, the nucleus is absent here. So this absence of the nucleus is a feature of what? Prokaryotes. So if you talk about the prokaryotes, they lack the true nucleus. So this erythrocyte RBCs, they have the feature of both the eukaryotes as well as the prokaryotes. So RBCs, they are present in humans, and humans are multicellular organisms. Okay, these are multicellular organisms, means they are eukaryotes. And usually eukaryotes, they have a true nucleus, but here RBCs, they lack the nucleus, which is a feature of the prokaryotes. Clear? So what should be the answer here? Neither eukaryote nor prokaryotic cell. Let's have a look at the explanation. Now the mature erythrocytes, they are eukaryotic cells that have undergone loss of their nucleus as well as the organelle. Okay, so prokaryotes, what is the feature of prokaryotic cell? They lack the nucleus as well as the membrane-bound organelles. So initially, these RBCs, they possess a nucleus which is lost during the maturation. Now, if we talk about the prokaryotes, they lack the true nucleus. Okay, while the eukaryotic cell typically possess a nucleus, but RBCs do not possess this nucleus. So we can say RBCs have both the feature of eukaryotes as well as the prokaryotes. The next question is, the removal of the calcium from freshly collected blood will, okay, then what will happen? If you remove the calcium, options are result in the clotting, it will prevent clotting, option C is prevent oxidation of the hemoglobin, or option D, it will cause hemolysis. So clotting, we have discussed about the process of the clotting. So clotting is done with the help of the blood platelets, and this calcium ion is important for the blood clotting. So this is the important ion that is involved in the blood clotting. So if you remove the calcium ion, then the blood should not clot. Okay, now let's have a look at the other option. Prevent oxidation of hemoglobin. Alright, so oxidation of hemoglobin. So whenever, so hemoglobin is usually present in the reduced form. Okay, hemoglobin is present in the reduced form, and in reduced form, it can carry the oxygen. But a small part of the hemoglobin, or small percentage of the hemoglobin, it is present in the oxidized form. So whenever there is oxidation of the hemoglobin, then it results in the formation of methemoglobin. So the oxidized form of hemoglobin is known as methemoglobin, and in this condition, it is not able to transport the oxygen. So methemoglobin, it cannot carry the oxygen because it is oxidized now. Only the reduced form of the hemoglobin can carry the oxygen. So this is also not the correct answer here. Calcium is not involved in the, or it doesn't prevent the oxidation of the hemoglobin. Now last option is cause hemolysis. What is hemolysis? Hemolysis is the damage or the destruction of the RBCs. Destruction of the red blood cells, also known as erythrocytes. So what should be the answer here? It prevents clotting. Okay, what is the explanation? Now clotting requires the calcium ions, which is why the blood banks, they utilize a chelating agent to sequester the calcium present in the donated blood. And this prevents the clot formation. So a chelating agent is used. What is the chelating agent? It binds with these ions. So what are chelating agents? What is the role of the chelating agent? It binds with the metal ions. And upon binding, what they do? They stabilize the complex then. They stabilize the metal ion so that it will not react now. So after stabilization, this metal ion, this calcium ion, it will be stabilized and it cannot be used in the process of the blood clotting. So chelating this calcium ion or removing the calcium ions from the blood, it will prevent the clotting of the blood. The next question is, in the cardiac cycle, diastole is. So we have already studied when we were studying about the structure and the flow of the blood in the heart. We studied about the systole and the diastole. So diastole is what? It represents the relaxation of the heart, or you can say relaxation of the heart muscles. This is known as diastole. And what is systole? Another one is the systole. Systole is the contraction of the heart muscle. So this contraction and relaxation, okay, generates a heartbeat. Clear? And it also generates the blood pressure also. So one is systolic blood pressure that develops or that is generated by the contraction of the heart muscle, and diastolic pressure, it is generated when the heart muscles relax. So let's have a look at the options. First option is, okay, so the first option is the number of heartbeats per minute. Second option is the relaxation period after contraction of the heart. Option C is forceful pumping action of the heart. And option D is contraction period after relaxation of the heart. So what is the answer here? The relaxation period after contraction of the heart. Okay. Now let's move on to the next question. One of the difference between blood and lymph is. We have already studied about the lymphatic system. In that, we studied about the difference between the blood and the lymph. So these are the options. Blood has RBCs and WBCs, while lymph has the lymphocytes. Now, next option is blood has RBCs, while lymph has no WBCs. Option C, blood has WBCs, while lymph has RBCs. Option D is blood has dissolved organic salt, while lymph has no such inorganic salts. Now the correct answer is option number A. Blood has RBCs and WBCs, while lymph has lymphocytes. So when we were studying about the lymphatic system, we studied that lymph is devoid of these RBCs as well as WBCs, but it contains lymphocytes as well as the antibodies. So these lymphocytes and antibodies, these helps us to fight against the pathogens, or you can say it helps us to fight against the foreign antigens, and it kills the germs and thus protects our body. Clear? Now next question is blood vessels carrying the blood from lung towards the heart. Okay, through. So what are the options? Pulmonary artery, pulmonary vein, coronary artery, and none of these. Okay, so we have studied about these when we were studying about the structure of the heart and the flow of the blood within the heart. So pulmonary artery, it carries deoxygenated blood. So usually arteries carry oxygenated blood, but pulmonary artery, it carries deoxygenated blood. Okay, it carries deoxygenated blood, and it carries blood from heart towards the lungs. Whereas pulmonary vein, so pulmonary vein carries the blood from lungs to the heart. Okay, so veins usually carry deoxygenated blood, but pulmonary vein, it carries oxygenated blood. It carries oxygenated blood from where? From lungs. So oxygenated blood from the lungs is carried towards the heart. And in the question, what is he asking? We have to name a blood vessel carrying blood from lungs towards the heart. Okay, so what should be the answer? You should know answer is pulmonary vein. Now coronary artery. So coronary artery also carries oxygenated blood. Okay, and there are two kinds of coronary artery, right coronary artery and left main coronary artery. And they are made from the root of the aorta. Aorta is the largest artery present in the human. So they arise from the root of the aorta, which carries the oxygenated blood. And there are two kinds of coronary artery. One is the right coronary artery. Okay, so this coronary artery, it provides blood to the right and the right atrium and the right ventricle. So one is the right coronary artery and other one is the left main coronary artery. So there are two kinds of coronary artery that carries the oxygenated blood. Right coronary artery, I've told you, it carries the blood to the right atria and the left right atria and the right ventricle. Right atria as well as the right ventricle. Clear? So what should be the answer here? Pulmonary vein. So this pulmonary vein, it carries the oxygenated blood from lung towards the heart. So this was the end of this session. Hope you enjoyed it. And we'll talk about rest of the topics as well as some of the other questions in the next session. Thank you very much for listening.